Assalamualaikum warahmatullahi wabarakatuh. Today we are going to presentate a video. I'm going to make a video presentation about the presentation that we have that group 3 have already made previously, in which we are going to present a journal about the effects of differential gene expression on coding sequence feature with the analysis by one-way ANOVA. So the problem statement. First, we are going to tell the problem statement of this journal, which is to study and to examine the effect of gene expression on the coding sequence in S. cerevisiae genome. And to understand it, we must understand one-way ANOVA, in which it uses the null hypothesis, in which there is no significant difference in the population distribution, and in this case, the gene expression levels among the different CDS coding sequence feature of moderately expressed gene. And the next one is the description of variable, in which there are two, which is dependent variable, in which it means that the variable is uh, dependent on many factors, external factors. For example, like uh, the variable can change, the R test can change if we, for example, forgot to study or like that, and so it changes and does not be the same. Independent variable is a variable in which there will be no change at all, even if there are external factor, for example, the age or when we were born. So for the test conducted, uh, we use the data of 3,868 genes, of which 3,768 have associated coding sequence feature measurements. Protein coding sequence from S. cerevisiae, which are 5,863 sequences, were downloaded from the International S. cerevisiae Chromosome Sequencing Projects. Each complete protein coding sequence was analyzed for the specific CDS feature of codon usage, amino usage, and nucleotide composition using codon. In this research, a one-way ANOVA test was used. One-way ANOVA is typically used when you have single independent variable or factor on your goal is to indicate if variation or different levels of the factor have a measurable effect on a definite result. And as you can see, in this one-way ANOVA, there are 4 groups. And in this study, we controlled for group size by organizing groups such as that the number of genes in each group differ by a maximum of one sequence. In this method generates 19 groups of gene expression level, with each group containing approximately 200 genes. In order to find what if any coding DNA sequence a feature differs significantly between groups, we performed a one-way ANOVA, and the effect of gene expression on the coding sequence in S. cerevisiae, producing several interesting results. And first result in this graph are the base frequency of cerevisiae. And as you can see, there is no significant uh, difference in the mean frequency of GC, and which means that this frequency does not reject the null hypothesis. Same thing for the other one, base frequency, but C and T ending codons appear to be more translationally optimal than A and G codons, as you can see above. And the nucleotide frequency at the center site was plotted for all group genes. Now for the next one is the codon usage bias, measuring the coordinates on CR axis. And as you can see in this graph, we will see the effects of the codon usage in our sample. And as you can see, there is not much big changes that happens when codon usage is added, with codon is added in the gene expression. with minor change probably in the CR1. And it can be also said the same about the amino acid composition, in which our gene expression is added with amino acids. On the AA axis, as you can see, there are no significant changes. So for this discussion, this analysis clarified that no strong association exists between coding sequence feature and protein expression level in the majority of genes. And this observation confirms that differential gene expression levels significantly affect only a subset of gene S cerevisiae genome, and that biased nucleotide composition caused by the accumulated effect of mutational bias is the major determinant of codon usage and amino acids usage in non-highly expressed genes. Sorry for the... Okay, so for the conclusion, we can conclude that gene expression level does not significantly correlate with the coding sequence feature of moderately expressed genes. But on the other hand, our results are entirely consistent with the idea that highly expressed genes may be identified based on their codon usage, as you can see from the example of the CR1. The CR genes, that was affected by the codon. And thus, in this journal, this sample does not reject the null hypothesis, which claim it does not reject the null hypothesis, which claims there is no significant difference in the gene expression levels and among the different coding sequence feature of moderately expressed genes. And thank you for the attention. Mohon maaf jika ada kesalahan. Wassalamualaikum warahmatullahi wabarakatuh.